Hey, good evening. This is Mr. Benning, and I'm going to hit this third lesson of science. We're looking at the methods of science. And so we want to learn about the method about which science is performed. So you can describe, we're going to be able to describe the difference between observation and inference. You can look at what a control, an independent variable is, and a dependent variable is. And you can identify the scientific methods are the scientific method or methods that we're going to talk about a biologist can use for research. So it all begins with asking a question. And all questions that science does comes from an observation. And an observation is basically what I see. So an observation is what we see, and it's usually done in an orderly manner. So we're looking at an observation, we hear it, we see it, we do it in an orderly manner. And from that, we can make an inference. It's like your first basic hypothesis. So a hypothesis or an inference is basically an answer based on only one observation. Only data you have, you can make an inference. It is based on one. And so when we look at this, a scientist can make observations. Where can a scientist, or the different places we can do science, or like what we're looking at for science. So the science here is, okay, science can be done anywhere. I mean, literally anywhere. Science can be done literally anywhere. I can do observations outside, inside, in the woods, at the pool, at wherever you are, you can do science. And based on what you see, you're collecting data from your eyesight, and you're actually making conclusions, making inferences about what's going on. And when there are more observations, when more observations are made, we form a hypothesis. A hypothesis basically is an explanation that is testable. So like I can create a test based on an explanation that I give. Like for example, let's say, for example, I can make a hypothesis. Okay, if I put water on the stove and turn the heat and add heat to it, it will boil. I can test that. I can go get a pot, go put it on the stove, turn the heat on, and let it and watch it, and it will boil. That's a hypothesis, but it's testable. We can actually create a test to verify what I'm stating. Now, I want to get to some of these serendipity moments. What serendipity is a scientific discovery that is made by accident. Okay, so sometimes that we make a discovery kind of outside that just by chance, and that does happen. So a serendipity thing One of them is using isometric, or meaning just resting or just crunching our muscles. So they're using isometric exercise and blood pressure. Another one that I like to talk about is penicillin. That was discovered by the isometric exercise. One scientist decided to use isometric exercise to help train astronauts, and he found that it lowered blood pressure just by doing that. So he went about and tested that on regular people, and it worked. Penicillin is another one where basically a guy was looking at bacteria and under a petri dish, had his bacteria left out over the weekend on his desk, was eating lunch there, had a cheese sandwich, it molded, some of the mold spilled onto the petri dish, and what he noticed is when he came back in was that all the bacteria around where the mold had gotten to had been killed. That's an accidental discovery. That's how we got the whole field of antibiotics coming out. So, again, we're looking at sometimes you get lucky with this stuff. So, once we have our hypothesis or something that is testable, we want to collect data. So there was a whole process in the data collection. And the method that we use is called an experiment. An experiment is basically an investigation in a controlled setting. So it's an investigation in a controlled setting. And so we control a lot of things inside that thing. So a control group. So one thing that we have to have is that when we're doing like groups, is that we look at, okay, so we have to have a group that we do nothing to and a group that we do something to. So when we're dealing in population, especially this is used a lot in biology, a control group is a comparison group. So this is like we've done nothing to them and are for comparison. The experimental group, on the other hand, we've done something to them, and we compare those to the control group. And so one big thing with this one is drug studies. So what they do is they'll take a drug, they have a new pill out, and they give half of them the pill with the drug in it, and half of them a pill that has no drug in it, that has everything else but the drug. They just want to look at if there is a psychological effect. And so they're just looking at comparisons here. So they're doing something to both groups, but one has the tested thing, the other one doesn't. And they look at comparisons. And so in the experimental design, we have a lot of things that we look for. So when we design an experiment, we have to determine if there is a linkage. So like if there is something that causes something else. So we'll control a lot of variables except one. And we're looking at how the output variable changes. So we have some comparison. So the independent variable is what the scientist changes, and it affects the outcome. So the scientist changes this, and it affects the outcome. Whereas the dependent variable is what is actually the outcome. So the dependent variable depends on the independent variable. And generally, the independent variable, when we do a lot of graphing, or if we do any graphing, the independent variable is the x, and the dependent is the y-axis. So it's kind of like, okay, if I want to look at, okay, which one's independent, which one's dependent, almost always the independent is the x, the dependent is the y. So when we collect data, we end up collecting two types of data. Sometimes we picked up data, we want to look at qualitative data, meaning it is a quality that is like color. Another one would be that's the big one that you look for is color. See how the color changes, or some sort of inherent quality. Like I'm thinking like, that's quantitative. A lot of things I can think of are quantitative, like qualities of the thing. Shape is another one. Maybe the shape changes. Maybe another qualitative data would be attitude. Like, I mean, that's something that's a quality. You can't really measure it, but it's something that we kind of look for. And then you got quantitative data, which is just basically your numbers. Okay, so sometimes scientists takes data that's numbers, which is most of it, and other times they're just looking at different qualities that they're looking for. So sometimes science is not just gathering data, but they're doing investigations, meaning they're looking at things like behavior, which is a qualitative thing, or even like going out in the woods looking for a new species. And they're not quote unquote collecting data, they're just trying to discover something. Okay, so once we gather the data, what scientists do at this point is they analyze the data. So I'm giving you a graph that just says basically the average daily temperature. You can see the highs and the lows, how they kind of go on. What do you think is going to happen next week? If I keep adding on, let's go on to Saturday. Do you think Saturday is going to be warmer? How much warmer? Is it going to be cooler? A lot of things I don't know, but just based on what I see, temperature and day, what's going to happen on Saturday? What do you think is going to be the next week, the next day? So there's a lot of things we can look at. Me personally, I can see it trending upwards, so we're probably on Saturday looking at an increase in temperature. Next week, we're talking 3 or 4 days. I don't know. I don't have enough data to kind of look at it. So I really can't make much of a prediction that far out. At this point, any prediction may be right, may be wrong, maybe some right, maybe some wrong. But that's the whole beautiful thing about analysis, is that when we look at data, we have to actually interpret what it looks like. And then once the scientist does that, it reports it. We have to report it. So we have to so a scientist will write a paper. Now, when I did my reporting when I was in college, I wrote a 20-page paper over and an investigation that I did and some research that I did. I will not expect you to write little, if anything. I might write maybe upwards of a paragraph of what did you notice? What did you think? If we do any lab reports, so just be aware of that. Be aware that we probably will not do investigations as an actual biology thing. We may do some investigations student-wise that I can do from a computer, maybe virtual. We'll look from there. But no, that's all I've got today. Have a good evening or good night or whatever it is, and I will talk to you later.